Make sure to watch the first lecture and make sure you really understand what's covered in the first lecture before you move on to the next lecture, before you move on to the next lecture. Just make sure, don't procrastinate, don't let those build up, don't wait till the last minute to do those. Because this material, again, kind of, it can build up, right? So if you don't procrastinate, you give yourself time for each of those lectures, you'll be fine. But we broke it down, I think, into, I'm really proud of what we did for cellular respiration as far as those lectures. I think that it works pretty well that way. So I probably won't ever change the way, unless I get good feedback from you guys saying otherwise. I think that this is the most understandable way to break it down, is how we did. So, but I definitely happy to hear input from you. Aside from that, I will kind of go through today what you can expect this week. I do want to announce that this week I would appreciate it if you could plan on coming to lecture all 3 days, so Monday, Wednesday, and Friday, if possible. I understand if your lecture is, you know, your prerogative to make it. However, I do plan on having, holding class Monday, Wednesday, and Friday this week. So if you want to come, come. And yeah, so I'll make sure to leave some time for questions at the end today, but as usual, you know the drill. If you have questions, raise your hand as we go through the lecture, or just unmute yourself and speak out, whatever you want, whatever you're comfortable with. Okay, let's go ahead and get started. What do you think? All right. Yes.
Just before we start, I want to ask a quick question regarding the cell communication like group discussion. How many people's posts do we need to comment on?
Just one. Yes. One. Thank you for asking that question. So you will notice that the cell communication wasn't put into small groups. And so it's basically everybody in the class posting to the same discussion board. So just comment on one other student's submission. You don't need to comment on multiple people. So just comment, try to find somebody that hasn't had somebody comment on their cell communication post yet, but just one other person. Yeah, you don't need to do more than that.
Thank you.
Yes, thank you for asking. All right, so let's take a look at what we've got before we get into muddiest points. I did want to make sure that we can address finally muddiest points, the soap and membranes. That's something really important that I want to comment. And then we'll go into muddiest points from last week. Last week was one of our, I would say, we have talked about, we have, covered sort of like the macroscopic scale for this class. We have talked about like cell structure and organelles, and now we're really talking about cell function, but that is kind of focusing in. So cell signaling was one of those first sort of more abstract topics that we cover in this class. Cell respiration, which we're covering this week, is very similar. Next week, we'll continue with metabolism, and then we'll kind of continue with some biochemical processes. So I think that you guys did phenomenal on cell communication, understanding it, but I do appreciate that there are some things that we need to go into depth about. So we'll spend some time on that today. Before we do that, I do want to go through a little bit of what to expect in Bio 181 this week. Again, if you can, please plan to attend lecture Monday, Wednesday, and Friday. I'll have activities every day this week. Friday is a little bit special because I'm inviting somebody in to talk about something I'd like to set up for this class. So if you can make it on Friday, that'd be great. And as well as that, something that I really want to mention is the Unit 2 review is open. It opened last Wednesday. It will close this coming Wednesday. When I checked last night, only 16 of you had started the review. So this is a lot of material for Unit 2. Remember, it covers stuff from both Unit 2 and Unit 1. And so it's cumulative. Do not put off studying for the checkpoint until the last minute. Okay, the checkpoint has way more material than the first one because it's cumulative. So make sure you're studying for that. The review is an excellent way to study. Do not plan on doing all 3 attempts of your review on Wednesday and then trying to do the checkpoint, like right after that. Give yourself time to process and understand the material, or it's not going to stick. So only 16 of you had started it last night. I hope when I check this evening that number is closer to like 100 of you having started the review. I see a question. Let me see. Let me get out of this. Nort, yes.
Okay, so I just had a question about the unit review. So I took my attempt this weekend, and then when last time I took it, it was like I took it once, and then I got new questions to do it this next 2 times. This time I took it once, and then the second time, since I didn't get 100% the first time, it just showed me the same exact test.
Yes, sorry about that. That has been fixed.
Okay, so I can take it the third time?
You can take it a third time, and actually I gave everybody a couple extra attempts because of that issue. So you should be able to take it more than one time more and see.
But I'll still keep my 100% for the other times?
It's still the best score.
Okay.
Yes. Yeah, sorry about that. Thank you for bringing that up.
Thank you.
For those of you who had taken it and took more than one attempt at it and noticed that the questions were the same, that has been fixed. So now every time you take it, you will see new questions, and your best attempt will still be the one recorded. So, but again, thank you. Make sure you're not waiting till the last minute to do those. Those are super helpful for the checkpoint. Speaking of the checkpoint, that will open Thursday at 12 a.m. and it will close the following Sunday. You have 1 attempt at the Unit 2 checkpoint. It is timed. I'll be able to give you the time for that on Wednesday. It will be 60 questions this time, so 50 questions from Unit 2, 10 questions from Unit 1, so it's the cumulative portion of it, and still 100 points total. So 100 points coming from each of those. So basically, each of these will just be a little bit over 1 point, or a little bit under 2 points, I should say. Aside from that, you do have a couple other things to expect in lecture this week. So hopefully everybody has submitted your rough draft of the e-portfolio for cell communication. If you have trouble posting it to the discussion board, just send it to me via email and I can help you get it posted. And make sure you peer review just, again, one other student's submission before Wednesday. Ideally as soon as possible. And then your final draft of your e-portfolio assignment is due Sunday, so this coming Sunday. So for lab, individually, so I couldn't add, this is not yet posted, but it is posted now. You do have another Labster this week. You have until Sunday to complete this Labster. So this is similar to the cell communication, sort of like lab simulation. It's just over cellular respiration. I would recommend not attempting this until you get through this week's lectures. So make sure you watch the lectures, take your concept checks, and then I would do this kind of like towards the end of the week, either Thursday or Friday would be ideal, because having the lecture content will be very helpful for completing this. One thing I want to mention about Labster is that I posted their tech support information in Canvas under the tech support module, but their tech support, unfortunately, unlike Connect, their tech support is only open Monday through Friday. And so I would really, really recommend working on your Labster simulation prior to the weekend, so that over the weekend, if you encounter technical difficulties, you can still contact tech support about those. So, but yeah, you have, just like last week, you have 2 attempts. Your best will be recorded. You have to complete 100% of the simulation. But yeah, so over cellular respiration. I think I have that due on either Friday or Sunday, But again, please try to make sure that you do it before the weekend so that you are not stuck over the weekend without tech support. As well as that, everybody needs to watch the enzymes lab video individually and take the enzymes pre-lab quiz. So this originally was supposed to have a due date of today, but I pushed it back until tomorrow at 11:59 p.m. So you have until tomorrow to do the lab video and the lab pre-lab quiz. So that's what you're doing individually, so for sort of like lab stuff this week. As a group, you should meet at your scheduled lab time. And as a group, you're going to be working on the enzymes kinetic lab and then completing parts 1 and 2 of that lab. So similar to osmosis, where you did a couple of weeks focusing on a lab, working on your writing skills concerned with that lab. Similar here, so we're really focusing on writing skills and making sure that you have plenty of opportunity to work on writing. This will be a little bit more intensive than it was last time, only in the way that we are going to make sure that you get more one-on-one feedback with your lab instructor before you turn this in. That will be explained by your lab instructor. And just to make sure, again, that you get really good feedback on your writing before you submit it and before it's graded. We just want to make sure that you have that face-to-face communication going on. So, yeah, all right. So that's kind of what to expect in lab. As usual, all of it can be found in our Canvas shell. So this is what our homepage looks like for this week. Week 8 module, kinetics lab. This is your recommended schedule. I hope everybody saw my announcement that, you know, just remember that when it comes to this recommended schedule, it is recommended. The due dates, the definitive due dates for these assignments are all listed in the actual modules. So you can see the reading assignment. Even though I recommend doing it, I think I recommended it for you to do tomorrow. It's not due until Sunday. So that recommended schedule is just to help you kind of with time management. So if you're curious about actual due dates, use that to-do list. But I would really think the best way to do it is just to look in modules and look at the due dates under the actual assignments. That will tell you definitive due dates. And there's your Labster. So yeah. Okay. Are there any questions before I move into lecture topics from last week?
I had a quick question.
Yes.
And so if we ever have any questions about the assignments or labs or anything, where can I get in contact with you? I emailed you a couple times, but like I don't know what to do.
I know, I'm so sorry. I saw your email. I'm sorry I haven't got back to it. My daughter got sick and then I got sick and then we're just finally recovering.
Totally fine.
But I did see your email and I will be responding as soon as possible. Yes, email is the best way to get a hold of me. But with lab grading and stuff like that, the lab instructors are really kind of where a lot of that stuff has to happen, but I can totally give feedback. I just don't have a lot of tons of say into that grading. But if you want to get input from me, definitely like email is excellent. Office hours, excellent. And I meant to get back to your email. I just hadn't had a chance to do so yet.
Okay, got it. And also for office hours, when is your office hours available?
Yeah, my office hours are Monday, Tuesday, and Thursday, and they are from noon to 1. But if those don't work for you, you're more than welcome to just send me an email and set up hours outside of that.
Okay, got it. Thank you so much.
No problem, thanks. Noor?
Hi, I just wanted to ask how your daughter's doing.
Oh, thank you so much. She's good. I don't know really what's going on. She just, you know, she's not like horribly sick. It's not, you know, it's not, I'm very grateful. It's not like COVID or anything, but she just, every time she eats, she feels really nauseated and I'm not really sure what's going on. I'm sure it's just like a kid thing, but I don't know, it's exhausting.
How old is she?
She is 4.
It's so cute.
Oh my gosh, I'm completely biased, but she is the most precious thing I've ever seen in my life.
You should bring her to a lecture next week.
I will try to have her come and say hi to you all. I will definitely do that. She's so sweet. She would love that. That would be her dream come true. Yeah, I'll try to do that. She loves science and stuff. So, but yeah, I will try it. Yeah, it's the thing with kids though is that they're always sick, and oh man, her little tummy issues lately have been a lot. But that's okay. She'll grow out of it, I think. Thank you for asking though. All right. Okay, so let's get into some lecture stuff. What do you think? I'm excited. I have to say, I'm again, when I was going through the lecture content and the Muddiest Point discussion boards from this week, I have to say I was so proud of you all because cell communication is a tough subject. And I know when I was in college, I really actually struggled a lot with cell communication. And when I was looking through the discussion board, it was really, really wonderful to see a lot of you say, I felt pretty comfortable with the majority of this. That made me really happy. So, you know, keep up the good work. I'm proud of you. That it's a tough subject. So the fact that a lot of you were like, I feel like I have a good handle on this made me really happy. The stuff that you did struggle with is just like very confusing stuff. So let's talk about it. I think I'm going to come back to this on Wednesday. I know I keep pushing it off, but I want to jump into signal transduction. So I do want to just mention something that's very important, which is that signal transduction and cell communication are sort of interchangeable terms. So when you talk about cell communication and signal transduction, there are 3 steps. And the first is receptor activation, where we have a signaling molecule or a ligand bind to a receptor. So that's receptor activation, a ligand binding to a receptor. And then step 2 is technically called signal transduction. And that's just where that signal is sort of like communicated through the cell. So this is like localizing the signal to a certain place. So maybe the nucleus or an organelle or even in the cytoplasm. The purpose of a signal transduction pathway is basically just to get this message through the cell where it needs to go. All right, it's a series of chemical reactions. And then the final step, which is 3, is the response. So you have the signal, the communication of the signal or signal transduction, and the response. So essentially you can think of it as like you're calling somebody. So the receptor activation is picking up the phone. The signal transduction is speaking into the phone and that message kind of going to the other person. And the cellular response is that person like hearing the message and responding to that message. So the responses are very varied. A lot of times the responses can be a reaction mediated by an enzyme. It can be changes in cell shape, so using structural proteins, or as we'll talk about, I think in like 3 weeks, we'll talk about genetic changes to the cell, which is probably one of the most common kind of changes that you'll get. So it looked like when I looked through the Muddiest Point discussion board, one of the most confusing topics was the mechanisms of cell communication. So direct versus contact dependent versus autocrine or paracrine and endocrine. So I wanted to talk about that. I think that talking about this in analogies is a really good way to do it. And that's still like to this day how I think about these things. So I tried to do that. So the first mechanism, oh, I see a question. Let's see. Yara, hello.
Hi, I was just wondering if you could turn on captions by any chance.
They should be on. Are they not showing up?
No.
Hi, Nora. They're not showing up. I have them on.
Do I have to like turn them on specifically?
You should have to. I'm not sure, but I have live transcription
Is
on.
it the live trend? Do I have to turn it on my end?
Yeah,
Yeah.
I can see your live transcript.
Okay, okay, I got it. Thank you.
Oh, okay, no problem. Sorry about that. Okay, I'm glad you brought that up though. All right, Tiffany, did you have a question? I think you're muted right now.
You know what? I was just gonna let you know that I could see captions on my end, but
Oh, okay.
I see
didn't.
she figured it out, so it's okay.
Okay, good. Thank
Sorry.
you. I didn't know that they had to be turned on your end. I apologize for that, but good to know. Okay. All right. So thank you very much. Yes, I will always try to, if you don't see live transcription, let me know. I want to make sure that's on. Okay, so for our types of cell signaling, the first type of cell signaling. So what I should mention is that when we talk about cell communication, what we are talking about is we're talking about receptor activation. So how are these cells getting messages, right? So essentially, like, where are these messages coming from, and how close in proximity are the cells that are receiving this message? So the first one is direct intercellular signaling. So anytime you hear the term, like the prefix inter, what you should infer is like it's inside or between cells. So with this, direct intercellular signaling means that this is happening between cells, not outside of a cell. Like literally, the cells are connected. And so what that means is these messages are spreading cell to cell through either gap junctions, so if it's animal cells, or plasmodesmata, if it's plant cells. So this would be cells in a tissue. So you can see here, this is a bunch of cells lined up next to each other. You can see these cells have gap junctions, and with direct intercellular signaling, the message is being spread, see, from this cell to cells that are literally attached. So remember when we talked about gap junctions, we talked about how this allows multiple cells in a tissue to receive like the same unified message. So this type of signaling is direct intercellular. So notice with this, the cells are connected to each other because they're connected by cell junctions, those gap junctions in particular. So that's intercellular signaling. Think of like inside the cell, spreading between cells. Okay, the next is actually one of my favorite types of cell communication, which is contact-dependent signaling. So similar to direct intercellular signaling, this is cell-to-cell communication, but the difference here is that it's not through gap junctions. So these are cells that are not connected to each other. So that's what differentiates the two. This is cells that are close in proximity, right? So they're very close to each other, and it does require the cells to touch each other, right? But the cells are not connected. So these messages are not passed through like cell junctions, through gap junctions. So instead, one cell has the ligand or the message, and the other cell has a receptor. So it's called contact-dependent signaling because it'll have two cells and they have to come in contact with each other to convey a message. So I like to think of this as sort of like a handshake, right? So you have two people, they come up and they shake hands, okay? They wash their hands first and they wash their hands second, and they're wearing masks. So they come up next to each other and they shake hands. So that's what this is. So again, big difference between intercellular signaling, where the cells are close in proximity, in fact, they're attached to each other, and they're sending a message through the cytoplasm, through gap junctions. Whereas contact-dependent signaling, these cells do come in contact with each other, but they don't have gap junctions. They're just sort of like shaking hands, right? So one has a ligand, the other has a receptor. It's just, again, this cell is trying to tell this cell something, right? So I want to mention a little bit more about contact-dependent signaling because I think it's very fascinating. It is personally one of my very favorite types of cell signaling, but I like to talk about its relevance in cell growth via what is called contact inhibition. So with contact inhibition, what contact inhibition is, so right, contact dependent signaling means that this cell is trying to tell this cell something, right? So remember, the ligand is the message and the receptor is what receives that message. In the case of contact inhibition, the message is stop growing, right? So this cell would be undergoing mitosis, and then this cell is trying to say, like, stop growing, okay? So this is really important. So what you're looking at here is a layer of epithelial cells. So this is skin, okay? So this would be a layer of skin, and you can see what happens is, let's say you get like a cut in your skin. So you can see this big divot. This would be a cut in your skin. What normally happens is after you get a cut, these cells start to grow closer and closer and closer together. And essentially, they just keep growing until they essentially grow enough to close the gap that was caused by that wound. But what will happen is that they grow only until they contact each other, right? So this contact-dependent signaling. So what happens is with this cell will produce another cell here, this cell will produce another cell here. They grow and grow and grow and grow until they touch each other. When they touch each other, they communicate to each other, stop growing, right? We only want to grow until we touch each other, and then we don't want to grow anymore. So what happens is if we don't have this type of signaling, right? So if cells get close together and they don't say stop growing, those cells will keep growing and growing and growing and growing, and that's what you're looking at over here, right? So let's say this is kind of, again, here, a layer of cells. You get a cut, right? And then the cells start growing. If when they contact each other, they don't tell each other to, like, stop growing, they'll start growing and growing and growing into a big mound. So you can see here, there's like a big mound of continuously growing cells. So if you can imagine that, right? So picture this. Picture you get a cut on your arm, right? So here's your wound, right? You have a big cut in your arm. The cells grow together to patch that cut. But what would normally happen is once they contacted each other, they stopped growing. And that's why your skin is sort of like the same, you know, like size, essentially. But what would happen is if those cells started growing beyond when they contacted each other, on that cut, you would have a big mound of cells. So what is a mound of cells that continuously grows despite signals that tell it not to grow? That's a tumor. So this is one of the mechanisms you can see in this picture. This is actually one of the things that happens with cancer cells or tumor cells, is they lose the ability, like if you have one cell being like, stop growing, this cell essentially is like, I do what I want, and it just keeps growing. And that's what you see here, right? So this cell should have got the signal to stop growing, but it's just not listening. And then it keeps growing and growing and growing, and now this is a tumor. So contact inhibition is a type of contact-dependent signaling. It relies on cells coming into contact with each other and communicating a message through contact, okay? I saw, Nora, did you have your hand up?
Yes, sorry.
Yeah, no, it's good.
Okay, so if you get a cut and then you get stitches on the cut, how does it grow? Like, what happens with that?
So that is what happens. So if you get like a cut and you get stitches, essentially what stitches are trying to do is so like one of the issues though with like a big cut is that it does leave your blood vessels and underlying tissue open to infection. So you have a lot of bacteria on your skin. And so one of the big purposes of stitches is trying to kind of like seal off a wound so that it's kind of like trying to prevent infection. However, you'll notice one of the first things that happen before your tissue starts growing together is you'll get the formation of scar tissue. But that scar tissue has to sort of fill in a space. So one thing that suturing does is it lessens the amount of space between basically where a wound is, and therefore it lessens the amount of scar tissue, and it kind of promotes tissue healing as opposed to just the development of scar tissue. So there are practical purposes beyond just like trying to eliminate scars. But one of the big things suturing wounds is to prevent infection, but also kind of limit scar tissue and promote tissue regrowth instead.
So if I were to put on like a Band-Aid on like a small but deep cut, would it still heal itself without stitches?
Oh, yes. Yeah. Not all wounds require stitches. And a lot of times, like, for example, like paper cuts are so painfully deep, but it's not like a big gap in that tissue. So a lot of times, again, it would depend on like the size of the wound, whether you need stitches or not. It's a good question. All right. Does everybody feel comfortable with, so far with the two types of signaling we've talked about? So with intercellular signaling and contact-dependent signaling, the differences between these. We feel good? I feel like those are two of the most commonly confused ones. So the big difference here, intercellular signaling, the cells are attached and communicating through gap junctions. And contact-dependent signaling, these cells are not attached, but they do signal via coming into contact with each other. So it's like a handshake. Right. Okay. And then we talked about contact inhibition, which I think is just one of the most fascinating methods of contact-dependent signaling. Okay, so let's move on to what's called autocrine signaling. And what you should immediately think of anytime you hear the term auto is self, like autobiography, right? So autocrine signaling is when you have close communication, no contact, right? So the two previous methods of cell communication we talked about did involve the cells contacting each other. With autocrine signaling, you don't have cell-to-cell contact. So you'll notice like here is the cell producing this message, and it's not in contact with any of these cells. But the thing that characterizes autocrine signaling is that this cell is producing the message, this little blue message, and this cell is also receiving that message. So it's a little bit hard to see in this picture, but autocrine is like autobiography. It's basically like, I say the analogy is like talking to yourself, right? So like, oh, I really need to study. Or like, oh, I really need to drink more water, right? So it's sort of like communicating a message to the cell itself. So another thing though is that sometimes, not always, autocrine signaling will involve this cell talking to itself, but also to its neighbors, right? So that is common as well. You'll notice here, like all of these cells look just like this cell. So in this picture, it's trying to convey like, this is an epithelial cell, and these cells are also all epithelial cells. So in this case, autocrine signaling means the cell is kind of communicating a message to itself, as well as all the cells that are similar to it, right? But again, I think when you think of like autocrine signaling, I would really recommend you think of like talking to yourself or autobiography, writing a book about yourself or about your life. Because with autocrine, that is the biggest characterization, is that the cell is communicating with itself. It produces a message and it hears that message. Again, sort of like, I need to drink more water. And then drinking more water. Right. Okay, so that's autocrine signaling. Paracrine signaling is when this cell is producing a message to nearby cells, but not to itself. So you see the big difference between autocrine signaling. It is not direct cell contact, so these cells are not like contacting each other. but it is very close in proximity, right? But again, with autocrine, the cell is communicating with itself and its neighbors. With paracrine signaling, this cell is communicating a message only to its neighbors, right? But again, this is a very close proximity. So it's nearby cells. So I would say that like the best analogy is like talking to others directly, like in the same room. So talking to somebody next to you, right? So you're not, it's like me being like, you need to drink more water. Not me, I'm hydrated. You need to drink more water. Right. So paracrine signaling is like this cell telling nearby cells what to do. The big thing here is that those cells are close by. So one of the big examples, and I think this was on the concept check, is what about like neurotransmitters and synaptic signals? So when it comes to nerve signaling and like whether two nerves are communicating with each other, or I should say neurons are communicating with each other, or if a neuron is communicating with a muscle cell, neurons don't actually contact the cells or tissues that they're signaling. They actually come into what's called like a synaptic space. And you'll see, so this is kind of demonstrating two neurons. They're not directly contacting each other. There's this little space between these. It's usually filled with like a fluid. But what happens is the signaling cell produces a ligand. You'll notice that this ligand is released by exocytosis. But the neurotransmitter is released, this signal, a ligand, is released into a space, and then it's received by a very nearby cell. So it's a close proximity, but they're not in direct contact. So that's paracrine signaling. This cell is telling this cell what to do. They are close to each other, but they're not in contact. Okay. Big difference here is that paracrine, this cell is telling this cell what to do. This cell is getting the message. This cell is not, right? So that's what distinguishes autocrine. If this was autocrine, this message would also be received by the sending cell. But paracrine is this cell telling this cell what to do. And then, oh, I saw, is there a question?
Yeah, I just have a quick question in regards to that slide. So
Yeah.
how do neurons then stimulate like muscles, you know? So is it the same through paracrine signaling or through like gap junctions? Because I know you mentioned that, you know, through gap junctions, you know, neurons stimulate, you know, the muscles. So
Yes,
that makes sense.
That does make total sense. So that's an excellent question. So neurons, let me see if I can find something here. Just 1 second. Let's see, nerve. Trying to find a picture that would demonstrate it well. Okay. So this picture does a pretty good job of showing like how a neuron would stimulate a muscle cell. So I'm gonna just for a second just screen share so you can see everything. So with a neuron stimulating a neuron, you have two neurons and you do have a space between them, and this is sending a neurotransmitter that is received by this. So it is paracrine because there's a space between the two neurons. The same thing is true for when a neuron stimulates a muscle cell or muscle tissue. So you can see here the neuron, and you can see it releasing the neurotransmitters. This is the muscle tissue, and you can see that these messages are still communicated, but they're still communicated through a space. So no matter what, neurons communicate through what are called synaptic clefts, which is that space between either a neuron and a neuron, or a neuron and whatever tissue they're going to stimulate. So that's how neurons would communicate with muscle. But the question you're asking is then how would muscle cells then communicate the message to each other? And that would be through gap junctions. So there's two levels of communication, right? It's the nervous system telling the muscle cells to contract, but then the muscle cells telling all of the muscles in that cells in that tissue to contract. As if you, it's a little bit difficult to see from this picture, but this neuron is communicating with just one muscle cell. But what this message has to do is it has to be communicated to all of the other muscle cells in that tissue. And so the first signal from neuron to muscle cell would be paracrine. But then from muscle cell to muscle cell would be intercellular signaling through gap junctions. Does that make sense?
Yeah, that makes perfect sense.
Good. That's an excellent question. So much communication. It's so cool. I love it. Okay.
Is that the same for cardiac tissue as well?
Yeah, it would be exactly the same for cardiac tissue. Cardiac tissue actually is composed of a type of muscle cells. It's just smooth muscle instead of, really, I can't remember. Smooth muscle instead of.
Is it skeletal?
Skeletal. Thank you. Yes. Smooth instead of skeletal. Yeah, good questions. You guys ask so wonderful questions. I love it. Okay. So excellent questions. So I think we feel good about paracrine. Again, it's nearby. So the big difference between paracrine and endocrine is that endocrine is communication of messages over great distances. So again, paracrine is when you're communicating a message that is close by, right? You're talking to somebody in the same room as you. Endocrine is distant communication. So like this cell is communicating a message, and that message is being spread over a long distance to a different, to a different cell. So the analogy would be like with paracrine being like talking to somebody in the same room as you. Endocrine would be like calling somebody on the phone. So like they're not in the same space as you. You're having to call them, and that message has to be communicated over a long distance. So yeah, so kind of, I think that the best way to talk about like comparing these different methods of cell communication is just to think like, sort of like who is the cell communicating to? Is it itself or another cell? Are these cells in direct contact with each other? Or I guess what I should say is like, what is the distance that this message has to travel? So with like direct intercellular signaling, the cells are like attached to each other. So the signal is literally spreading through their cytoplasm. They're connected to each other. It'd be like, you know, like, I guess if I was pregnant and, you know, when I eat, that food is shared with my baby, right? That baby is in there, is getting the same food as me. So we are connected through our vasculature. You know, these cells are connected. They're connected by gap junctions in that way. Contact-dependent signaling, these cells are not connected, but they do come in contact with each other. Again, like a handshake that's communicating a message. Hello, I'm nice, hopefully. Right, so contact-dependent, this message is direct between two, but it's not through the cytoplasm. It's through a signal and a receptor. Autocrine signaling is close by, nearby signaling. And the big thing that characterizes autocrine signaling is that the cell is communicating to itself. It may also be communicating to nearby cells, but the thing that is characteristic of autocrine signaling is that it is communicating with itself, right? So talking to myself, I need to drink more water. Paracrine signaling is this cell communicating to other nearby cells. So that would mean being like, you need to drink more water, right? So nearby, same space, talking to your neighbor, but giving them a message. Whereas endocrine signaling is calling somebody, all right? Me calling, I guess my husband, I'm in Missouri, he's in Arizona, me being like, you need to drink more water. Okay, it's hot there, right? So that's kind of the difference. It's over distances. All right. So we have a couple of more minutes, but I don't want to get too much into this next one until Wednesday because I want to have time to talk about it. But what I want to mention right now is all we've really talked about is so far is kind of clarifying like where the signal is coming from, right? So importantly, we've talked about that the signal can come through the cytoplasm, right? Through gap junctions, in which case we would kind of just initiate this. It would come through, if it's talking to itself, this message would come from the cell communicated here. It could come from another close cell, right? So either via direct contact or paracrine, or it could come over long distances, in which case it's endocrine. But the important thing is no matter where the message comes from, what happens is once this signal binds this receptor, a series of reactions happen and those stimulate a response. So what I want to talk about on Wednesday when we meet again is what happens when that signal binds this receptor. So we have a couple different types of receptors. We do have cell surface receptors, so those are receptors attached to the cell membrane. But we can also have intracellular receptors. So those are receptors that are in the cytosol. But no matter what, they're going to communicate, undergo, or like stimulate, I should say, a chemical reaction pathway that results in a response. So really what we're talking about when we talk about the different types of cellular receptors is when this ligand binds this receptor, what happens to stimulate this response? Okay, so that's really what differentiates them. And we'll talk about those on Wednesday. My favorite thing about talking about that is that if you enjoyed the plant communication lecture and talking about how plants respond to pathogens, which it seems like a lot of you did, I really like talking about cell receptors because your immune system is really dependent on cell receptors and where they are. So I talk now on Wednesday, what we'll do is we'll talk about the different types of cell receptors, and then I'll give you a couple examples of how these are present in your body and how they're used for immunity. So sort of in the plant cell lecture, how we talked about how you can have pathogen elicitors and how the plants have receptors that respond to those elicitors. I'll tell you a little bit about how your cells respond to human pathogen elicitors, which I mean, that's my favorite thing in the world. So thank you for this opportunity. All right, so that is all we have for today. So we do have a minute or two more. I have to pop off in about 5 minutes, but I can hang out for about 5 minutes, but lecture is done. So if you want to go, that's fine. I understand. I will see you on Wednesday and also on Friday, and we will continue this lecture on Wednesday. If you have questions, again, I'll hang out for about 5 more minutes.
Dr. C, I have a quick question. For the
Yeah.
review, I had the same issue as Zenor did, where it had the same exact problems. However, I went back and it says I only have one try left.
That should have been fixed. Did you check it today?
Yeah, I actually, just after you said that, because as of yesterday, well, I think when I took it on Saturday, it said that I had no more tries, or because I got 100% on the second one.
Yeah.
So it wouldn't even allow me to look at the third one to see, you know, what type of questions they had. But when I clicked on it now, it's available, but it said third try, third out of 3.
Okay, I'll check that again. Can you send me an email just
Sure.
so that I will have it? I thought I fixed it, but yesterday it was having
That's
some
okay.
issues. It kept.
Well, let me go back in there to see the problems, or try it again for the third time. But I guess after this time, it's maxed out.
Yeah, I tried to make sure everybody that had taken their attempts had more attempts. So if you're seeing only one attempt left, send me an email because you're supposed to have more.
Okay, awesome. Thank you so much. I appreciate it.
Oh,
Thank you.
well, thanks for letting me know.
You're welcome.
Sorry about that.
Dr. C, I have a question.
Yeah.
So for the muddiest points, I was very confused about the Lapster, the simulation and how it worked. So
Okay.
instead of asking a question on the lecture, I asked a question about the lab, like a specific question. I was wondering if that's okay.
Yeah, that's totally fine. That's totally fine. If I will try, I'll look at it again, and I will
It,
try to make sure that I'll like try to pop it in there for Wednesday.
it's just that I'm struggling like a lot on the cell receptors, like the different kinds of receptors. Like I understood what you just talked about today, like, but those receptors, the different types of receptors, that's where I get lost.
We're gonna
Um,
talk about that on Wednesday.
okay,
That's what we're focusing on
so
Wednesday.
I wrote a couple questions on my notebook when you were talking.
Okay.
So when cells, like say you get a cut, right?
Yes.
How do those cells know when to reproduce? Like I know they know when to stop because they communicate, but like how do they know, oh, we got a cut, we need to multiply?
We will actually have a whole lecture on that later. So we're going to talk about, and this will be in a couple weeks, but we'll talk about the cell cycle. Cells are actually programmed to reproduce. So typically what happens, like if you get a cut, the cells will be stimulated to enter the cell cycle and reproduce. And they will do that until they contact each other. That's when in the response of a cut, right? And also most of your tissues. But what happens is if they get into the cell cycle and they start dividing, if they don't get that message to stop, they'll keep going and going and going.
How would they not get that message? Do they just have like bad receptors or they just don't want to listen?
That is very complicated. And that it will differ depending on, honestly, it differs depending on the type of tumor or the type of cancer, because that would be characteristic of a tumor or cancer. And usually it does have to do with like a defective receptor. So they are lacking the receptor that enables them to get the message to stop. Or it could be actually at the genetic level where they're not, so like they have the receptor and they're getting the message, but that message is not being effectively communicated. So it just depends on the situation.
Okay, I have one more question.
Yeah.
What's the difference between autocrine and direct intracellular signaling? Because like I know they signal each other like nearby cells.
So the
Or
big difference is that direct intracellular signaling would be messages through gap junctions. So through the cytoplasm, right?
okay, so they're two
Autocrine,
different cells.
yes, but they're like connected in a tissue. So like muscle cells,
Okay, is that how
right?
they like stimulate, like they do perform those functions when they contract?
Yes, all at the same time.
Okay.
And then autocrine is when a cell sends a message and gets that same message. So we didn't talk a whole lot about examples of this, but it like I kind of talked about how it'd be like talking to yourself. Like a cell is communicating a message that it needs to hear. Sometimes that can be like stopping growth, but we didn't go into that.
But how would that be like viable? Like when would a cell need to communicate to itself to do like something?
Well, one example is like if a cell is infected with a virus, the cell will secrete messages to itself that stimulates that cell to like die, actually. So it's only that cell, like if that cell has a viral infection, it has to create like a message to be like, I need to stop this viral infection. So autocrine signaling is communicating to that cell itself, but also to nearby cells. It's saying like, there's a virus in me. And that cell receives that message and does something, and the nearby cells do something as well.
Okay, thank you so much.
Yeah, good questions.
Hi. I know I asked a lot of questions about the last one.
That's good.
Okay.
Questions are good.
So today you went over cell signaling. So I feel like now I understand it more than the lectures, but do you think I need more in depth than what you covered today? Or is this okay?
That's the depth you need to know. Yeah.
Okay, so
That's
I'm
sort
good
of
for
like distance. Yeah.
So then what else do I need for the unit review from this unit besides cell signaling?
I would say what we're going to talk about on Wednesday will go, if you feel, I think I'm trying to think about your muddiest point. Your muddiest point was kind of what we discussed today, right? Or
Mhm.
was it the receptors? I think. Did you feel
Like
okay
my
about the
question?
receptors? Mm-hmm.
Well, I'm just wondering, like, overall, because I feel like when I do the lectures, I don't really understand. I understand like 20% of the content. So Muddy's points help me understand more.
Good. That's what I want. Good. So I would say, like, we'll talk more about on Wednesday the actual receptors. And I would say that's sort of like from cell communication, what you really need to know is like what we talked about today, and then the different types of receptors. So, like, the difference between like enzyme linked, GPCR, and what's the other one? And ligand gated. So
And then last week's Muddy's points, and I should be good for the unit test.
I think so, yeah.
Okay, thank you.
Yeah. No
Have a good
problem.
day.
You too, see ya. All right.
Dr. Z,
Yes.
I have a question about the review. So I did the review last night and a question popped up from like last unit and I put the correct answer, but it like switched up the answer and the same thing happened when I took it last
Last
chapter,
time.
last test. Yeah, so I don't know like what the correct answer is because like I put the correct answer and then it says it's wrong. So then the next time I see it, I put that answer, but then it says
Still
that's
says
wrong.
it's wrong.
Yeah.
Oh, good. That's very helpful. What I would like you to do, if that happens, screenshot it and send it to me so I know what question. But also, have you noticed that like when you get a question like that, that there's a button that says like report this question?
Mm-hmm.
Oh, click that. So always give them feedback that that's happening.
Okay, because I saw the question on the test also for the first test, and I'm assuming like that's the question I got wrong, because I
What
didn't know
was?
it was about steroids. Like, wait, one second, I wrote it down.
What is it? Sorry, I was shaking my, there was a motorcycle outside my apartment. I couldn't hear what you were saying. Oh my gosh. But there, you were saying, are you looking up the question?
Wrote it down. Oh,
Okay.
okay. So it says if a person's ability to form steroids in his or her body was destroyed, what function would be affected?
Okay.
And the first time I got it, I put the answers where hormone production in the endocrine system would no longer exist or occur. And it's like one of them, it's select all that apply. So I selected 3. And the second one was estrogen would no longer be produced. And then the third one was cholesterol levels would keep increasing and cell
So,
death would occur.
okay. So the two that the estrogen and the hormones are correct. So if like it would impact estrogen production because it is a hormone, like you said, and then it would also impact like hormone production overall. But it would not necessarily mean that you're producing more cholesterol because cholesterol is also a hormone, so are steroids. So you just would not be producing more.
Yeah, on the first attempt, the estrogen and the molecule hydrolysis were still there, but the answer that changed, it said the ability to process insulin for glucose uptake would no longer exist.
That is also true, right? I think that is also true.
So would it be the molecule hydrolysis and the estrogen and the insulin intake only, those 3?
I'd have to see the question. Let me go back in there and can you send me an email and say, can you look into this question? And I will look into it because I'd have to see the question to make sure.
Okay.
Yes. But I'll
Thank
look
you.
into it for you. I'm sorry about the confusion.
It's okay. I just feel like this is the question I keep getting wrong because I don't know the correct answer.
I'll look at it for you and I'll help.
Okay. Thank you.
Yeah. All right, everybody, I do have to get going. But if you have questions, reach out via email is the best way. And I will talk to you soon. Bye.